Hi, everyone. Welcome to 104th Stanford MedAI Group Exchange session. This week, we have Margarita Bintsi from Imperial College London here with us to talk about her work on multimodal brain age estimation using population graph learning. Margarita is a final-year PhD student at Imperial College London, supervised by Dr. Daniel Ruckert. Her interests include graph machine learning and multimodal learning, with a particular focus on increasing interpretability. She also interned at Microsoft Research and the NASA Frontier Development Lab, where she applied machine learning models to telecommunication and solar physics. Margarita, thank you very much for joining us today. Before we start, do you have any preference on how you would like to take questions?
I'm fine with whatever, either during or at the end.
Okay. So everyone, let's try to make this session as interactive as possible. And without further ado, let me hand it over to Margarita.
So, hello, everyone. I am Margarita. As it was already mentioned, I'm in the final stages of my PhD at the Biomedia Group at Imperial College London. I'm supervised by Professor Daniel Ruckert, and today we'll be talking about the work we submitted and presented at MICAI last year, which is on multimodal brain age estimation using interpretable adaptive population graph learning. I'm gonna share my screen. Is it okay?
Oh, yes, it is.
Okay. And I realized I should have done the intro now, but it's fine. And let's start. So healthy aging in the brain follows a specific pattern, and the volumes are changing in a specific matter. But in the case of neurodegenerative diseases, such as Alzheimer's disease, for example, some connections between neurons stop working, and then this leads to atrophy and a loss of brain volume. Thus, in the case of neurodegenerative diseases, there is an abnormal accelerated aging of the human brain. Therefore, the difference between the real age or the chronological age of the person and the biological or brain age of this person is considered to be a potential biomarker for neurodegenerative diseases. Recently in the literature, population graphs along with graph neural networks have been used for different medical tasks. I will explain shortly what a population graph is. So in a population graph, every person, usually every subject, is one node. And the imaging information are usually the node features. So all of these nodes, the subjects, are connected among each other based on some associations or relationships among them. And then the population graph, which is one whole, is used as input to different GNNs. And the prediction is based on nodes. So we have node classification or node regression. The problem is that in the case of medical tasks, we don't have a given population graph. Thus, we have to figure out a way to build this graph, to construct it. And there are two common ways in the literature. The first, the most commonly used one, is to have a static graph. So we have a population graph and we make a choice on the way we will connect the edges. It's predefined and it stays static throughout the training. And it could work well if we had the graph, but it's usually not given. And a not optimized structure does not well, and it sometimes leads to performance even worse than a graph agnostic method. A possible solution to this is the use of adaptive graphs, which are graphs that are not predefined and remain static, but they are learned during training along with the training of the GNN. However, in the literature, in most cases, they do not take into account the non-imaging information, but they're mostly focusing on the imaging information, and usually they're not interpretable. And for the medical tasks and in healthcare generally, this is quite like important for the clinicians to trust every result. We said before, we mentioned that the static graphs, they sometimes do not work very well, and if a population graph is not optimized for a specific task, it may underperform even like a simple MLP. And this in the literature has been found correlated to the homophily, a metric for the graphs. Homophile graphs are the ones that have nodes with same labels connected to each other and not connected to nodes with different labels. While in heterophilic graphs, what we have is that the nodes are connected based not like only to nodes with similar labels. And this can work okay for some graph structures, like some GNNs that are more resilient to the graph structure, for example, the graph states, but there are some other like graph neural networks, such as GCN or GAT, for example, that are more sensitive to the graph structure, and they are affected highly by this. For this reason, in this work, we decided to work on adaptive graph learning and learn an optimized graph structure for brain age estimation. And we used an attention-based framework to do so. We used the graph laws that allows us training end-to-end both for the optimized graph structure and the GNN structure. And by using the attention scores, we were also able to create an interpretable graph in a way that I will explain later in the talk. We tested, evaluated our model on brain age regression, as we mentioned, but also we tested on brain age classification, which is something that is used commonly in related, like in the literature, so we could compare our results. Let's go to how we did this. So we trained an MLP that uses as input the non-imaging information that we are like interested, but also some imaging information. And then we gave attention weights, one attention weight for every phenotype. So it is global. And then we use these attention weighted phenotypes in order to build the population graph. The graph is sparse, so we don't have all of the edges. And we use this population graph as input to the GCN in order to have a prediction, either regression or classification. In order to be able to train this end-to-end, because we have a sparse graph, as we mentioned, and we cannot backpropagate directly as it is, we used these graph laws. Our loss has like two terms. The first term is for the optimization of the GCN, and the second term is for the optimization of our like population graph. It's basically for the optimization of the MLP, and then which will optimize the attention scores, and then this will give an optimized graph structure. But because, as I mentioned, we cannot do this end-to-end, we built these graph laws, we extended it to regression, using like a reward system. So we would reward edges that participated in correct predictions and penalize edges that participated in the wrong predictions. By having this combination of losses, we were able to train both.
Margarita, can I ask you a question here? So as I understand, this MLP is not being optimized beforehand. It's just randomly initialized and plugged into the system.
Yes, and for this, we are actually based on the assumption that not all the phenotypes are equally important for brain aids and for the construction of the graphs. So we train the MLP and we give attention scores to one for every phenotype. It's from 0 to 1. And intuitively, what we expected before was that the features that are the most relevant to brain aids and the most useful for the task would have attention scores close to 1.
Mhm.
The ones that are not relevant would have attention scores close to 0, and they would basically not used that much for the extraction of the graph.
So is it correct to assume that this MLP takes in concatenated features, non-imaging and imaging, concatenated features, and then it's also doing some kind of compression, like it's mapping those concatenated feature vectors into another space?
No, it takes the concatenated features
Mhm.
and then it produces one attention score for every feature.
Okay.
So what the output is actually a vector, which
Mhm.
is the size of the different phenotypes, which we used around 70. And then we have one attention weight for every phenotype, for all
And
of them.
how do you come up with these phenotypes?
The phenotypes. So we used the imaging phenotypes for the node features, then we wanted to use the non-imaging phenotypes only for the edges, finding the relationships. But this was not enough information for the graph structure to be built. So we did an ablation study. We combined, I have the results, we combined all of the imaging and non-imaging information and use them for the edges. Then we used only the imaging, only the non-imaging, or a combination of both. And we ended up using a combination of imaging and non-imaging information.
Okay, and when you say you used around 70 phenotypes, is it like 70 features, imaging and non-imaging combined, or is it something else?
Yeah, that's what it
It's
means.
Oh, okay. And I think we have a question.
No, it's 35. It's 35 for the edges. We have like around 70. I will mention it later in the data. We have 70 imaging features, but we don't use all of them for the edges.
Okay.
And we have around 20 non-imaging information.
Okay.
So for the edges, we use 20 non-imaging and around 15 imaging.
Okay. And I think we have a question in the chat. What is the supervision signal in MLP training?
What supervision signal?
In MLP training, what is the supervision signal? Like, how are you training MLP? I think that's what they want to know.
How we train MLP?
Yes.
Okay. So we train it using the graph loss term. So because we give some weight scores, and then we use the weighted phenotypes for the graph structure. And then we use the graph structure for the GCN. And if the results are good, then these edges are rewarded.
Mhm.
If they are not good, they're penalized. So the attention scores are updated accordingly. And the attention scores that give the minimized loss function are the ones that are given like the highest scores. The phenotypes are given higher scores. Does it make sense? We give attention scores,
Mhm.
and then we use these attention scores for the extraction of the edges.
Yes.
And then for these edges, if it works well, we reward the edges. If it doesn't work well, we penalize the edges.
And when you say you reward the edges, it's basically just rewarding the MLP. That becomes the supervision signal for the MLP.
Yeah.
Thank you.
Yeah. Okay. So yeah, we use the UK Biobank dataset because it has a variety of both imaging and non-imaging information. We used around 70 neuroimaging phenotypes from structural MRI and diffusion-weighted MRI, which were basically provided by the UK Biobank. We did not extract them. And then we also used some non-imaging non-imaging phenotypes, which are demographic, some biomedical, lifestyle, and some cognitive performance measurements. We had around 6,500 subjects, and we had a split for the training, the validation, and the test set. As I mentioned in the beginning, we focused on age regression, but we also evaluated for class classification task, because in other papers in the literature, they only tried the classification task, and we wanted to compare our results to them. The regression task is more complex. This is probably why it's not as explored, or the population graphs, not in general. And here we can see our results. We had some baseline methods. First of all, because we said that sometimes if the graph structure is just bad, it's not optimized, we have results even worse than a simple graph agnostic method. We trained a linear regression for age regression and a logistic regression classifier for the classification problem. Also, we had two more baselines, which are the static graphs. And the static graphs are basically what I mentioned in the beginning. And in our case, we used a similarity score for the imaging and non-imaging phenotypes, the same ones that we used in our method, but without attention weights. So we used all of them, a similarity score, and then we extracted the edges based on this. Actually, this is the static phenotypes, which is a combination of the imaging and non-imaging similar to ours, but we also used only the node features. And for the second case, we did not include non-imaging information at all. And finally, we compared our results to the DGM, which is the state-of-the-art method. It is also adaptive graph learning, but it only uses the imaging information, not the non-imaging one, and it's not interpretable. So as we can see here, for the regression task, we have the mean absolute error, and our proposed approach outperforms all of the others, the baselines and the state-of-the-art. As we mentioned, we see here that the static methods, especially the one with the non-imaging information, performs worse than the linear regression, which is something that we had in mind for the static graphs. The adaptive methods work better than the linear regression and both better than the static graphs, but our method outperforms the state-of-the-art. And for the four-class classification problem, we have a similar tendency, like the trend is similar exactly as the regression. A good aspect of the work, like an advantage, is that by having the attention weights, we are able to visualize the weights and then get an understanding, like have an interpretable graph in terms of understanding why two nodes are connected or not connected. So here we've visualized the weights for the top 5 imaging and the top 5 non-imaging phenotypes. So the ones that were given the highest scores by our pipeline. As we can see here, regarding the non-imaging ones, the pink ones, some cognitive performance measurement tests were given the highest scores. And also the stroke and like the blood pressure was important, but not as important as some of the imaging. And when we go to the imaging, some volumes were important and some measurements extracted from FA skeleton. Yeah, and the gray matters. The gray matter volumes were also important. These results are in agreement with the neurobiology literature. And as I wanted to mention that we had plenty non-imaging information. We chose to use some based on the literature and some work from James Cole, that they did exactly this. They had brain age regression, they had UK Biobank, and they tested different phenotypes to check which ones are the most relevant to brain age. And our results basically are in agreement with this work as well. So these were given the highest scores.
Margarita, you mentioned that abnormal difference between the chronological age and brain age is like one of the biomarkers for neurodegenerative disease. So
Yeah.
when you are comparing your results to the ground truth, so that ground truth? Like, what is that ground truth? It's the actual brain age of a person with Alzheimer, or just the
Yeah,
chronological age? Because if it is the chronological age, you will have a difference. If your model is working good, then you will automatically induce some error.
yeah, that's the thing with brain age. We don't have other labels than the real age that we know.
Mhm.
So what we do usually is that we train and test, like we do everything on healthy subjects,
Mhm.
and then we consider the brain age the same as the chronological, the real age. So in this work, we only use healthy subjects. We want to make the models perfect for the healthy subjects, and then when tested on
Okay,
healthy and on a Alzheimer's
patient.
patients, yeah, we can see the difference there.
Okay.
So it's trained on healthy subjects.
Okay, so all of these results that you're reporting, that these are for healthy subjects. So
Yeah.
did you do some analysis on like patients with neurodegenerative disease? Does it really show a huge difference between the
No,
predicted brain age?
for this work, we have not, because we
Okay,
don't have data.
okay,
But for the future work, we want to apply this for the ADNI datasets, in which we have more like healthy and Alzheimer's disease patients, because UK Biobank is mostly healthy subjects.
oh, okay, okay. Thank you.
I don't see the chat because I'm sharing. So,
So I'll let you know if I see any more questions in the chat.
okay, thank you. What we also provided is some these visualizations of the population graphs. So on the left, we have the static graph that we described before. So it's a graph, population graph constructed based on the cosine similarity of the phenotypes. And on the right, we have the same population graph constructed, but based on the cosine similarity of the weighted phenotypes, with the weights being the ones that provided by the proposed pipeline. Here, the colors are the ages, as you can see. The red are the old subjects and the blue are the young ones. And we can see that on the left, there is basically, there are no neighborhoods. It's a bit random. So like every node has a bit random neighbors, while on the right, there are clear neighborhoods. So the younger people are closer to each other, the older people are closer to each other, which is show us why the GCN probably works better in our case, because the GCN is highly affected by the neighbors. So if the neighbors are of different, like completely different age, then their node features, their imaging features are also of like different age. And when you have GCN, you average all of this information. So after 1 or 2 layers, you basically have no information about the age. While in our case, in the right hand, we have neighbors of similar ages, so their imaging features are also closer, and when combining them later in the GCN, we get more meaningful information, and thus we have a better performance. All in all, we proposed an end-to-end pipeline for adaptive graph learning, which is optimized. The graph is optimized for the task of brain age regression. We integrated multimodal information, which is like some imaging information and some non-imaging information. And we used attention scores in order to construct the graph, which also allowed us to increase the interpretability. We implemented this approach for node regression, but also for node classification for 4 tasks. And we outperformed both the baselines, the static graph-based, and also the adaptive state-of-the-art graph learning approaches. We were able, using the attention scores, to provide some insights into like the most important, the most relevant phenotypes for brain age and for the graph construction. And our findings were in agreement with the related neurobiological literature. As I mentioned, in the future, we want to evaluate this framework to other data sets and other tasks, like, for example, for ADNI dataset. And also what we would like to do is to have a CNN and extract features directly from the brain, so not taking the imaging information provided by UK Biobank, but extracting features from the CNN and then use features directly in the GNN and train this end-to-end, as we assume, like we expect that the features extracted for the brain will be the most suitable for the training of the GNN as well. And thank you very much.
Thank you very much, Margarita. So let's give our presenter a virtual round of applause. And let's wait if our audience have any questions. Yeah, so there's one question in the chat that is, in the reward function, what's the purpose of epsilon? Is it the same for all samples?
Let me go. So epsilon. Okay, here.
Yeah.
So epsilon is the average brain age. It starts by being the average. So we take all of the real ages of the people in the training set, and we have an average age. It is around 6 at first. And the purpose is that we have this reward function, and we want to see if this difference is smaller than the average at first. This does not stay epsilon, but later during training, it's decreasing. So the purpose is always to decrease this and this and this.
Oh, so the epsilon is changing during the training part?
Yeah, it's changing. I haven't written it correctly here. It starts by being the average, which is, this is the mean prediction that we will have if we did not train and if we gave the average age to everyone. And then as the training passes and our prediction is better, the mean is better, this is decreasing as well. So we can push the reward function more and more.
So initially it's just the average brain age of people in your data set, and then as your model trains, it's the predicted brain age, it's the mean predicted brain age of your data set, but it can also increase, right? Right, if it is dependent on the model, and as the model is learning, maybe it's learning to predict a higher mean age for the data set. Am I?
It could, I guess, but then the model would be wrong, like problematic in many ways before reaching this problem. If the mean absolute error increased.
No, I'm not talking about the mean absolute error. I'm talking about the epsilon only. Epsilon starts out by...
Epsilon starts at 6, which is the average age in this data set.
Okay.
But then is the mean absolute error, which is decreasing.
So the mean absolute error decreases, but does that, like, is the epsilon value remains fixed?
It's not fixed.
Okay. How is it updated?
It's updated, yeah.
How? The epsilon value?
It's the mean absolute error.
Okay.
Yeah, I should have written it more clearly.
That's okay. Thank you. Any other questions from the audience?
Yeah, I have a question. In this the graph that you've generated, the nodes are the patients, and these weighted features are the features of each patient. And how are you getting the edges? How are you calculating the weights for these edges?
Okay. Yes. Thank you for the question. So we have the attention weights. It's from 0 to 1. You can see my mouse. Sorry.
Yeah.
Yeah, I thought you couldn't.
I see your mouse.
Okay, so we have these attention weights, which are from 0 to 1. And then we have the weighted phenotypes. And then we have one value for every phenotype, for every person. And we used some similarity function to find the probability of two of them to be connected or not. We used like some distance function. We tried the Euclidean distance. We tried cosine similarity. I think we ended up going with the Euclidean because it worked the best. It's also an empirical, like an ablation test, an ablation study in this. So we had all of the probabilities. And then we used the Campbell.K trick, which is a stochastic relaxation of the KNN. Imagine like we used KNN function, but a stochastic relaxation of it. So we kept the top K neighbors for each of the nodes, which are the ones that are like the closest based on the phenotypes, the weighted phenotypes.
Okay.
Does it make sense?
Yeah, it does.
And is K also treated as a hyperparameter? Like what should be, how many top neighbors you should keep?
Yeah, we kept, I think, for the result 5, but it's just hyperparameter.
Okay, so Margarita, I'm still stuck up on that question of how are you getting these attention scores from MLP? What is how do you what is being back propagated? What is the label for the feature vectors that are you giving to the as input to the MLP? Can you please elaborate? I'm sorry, I couldn't understand
No.
earlier.
Yeah, it's fine. So we have some features, and then we concatenate some non-imaging and imaging and use these as input. And the output of the MLP is a vector of attention scores, one for every phenotype. Not one for every phenotype for every person, one for every phenotype. And then we use these attention scores to weight the phenotypes. And we use these weighted phenotypes in order like to find, and then we use KNN, the stochastic relaxation of it, to find the closest neighbors based on these weighted phenotypes. We use these KNN to extract edges. And then we use this graph structure as input to the GCN. But because this is sparse, so in every iteration, like every time, we have different edges. We don't have the same edges. So we cannot directly assign scores for every edge. What we do is that we use this graph loss that compares the prediction and the label, and we want to minimize the graph loss. So we want to maximize this. Yes. And by in every time we have like correct prediction, we reward the edges. And in this, every time we have a wrong prediction, we penalize the edges. So by penalizing the edges, we have it learns not to give high score to this attention weight. The MLP does not give high score to these phenotypes in the next epoch iteration.
Okay, so the L graph loss is being propagated end to end till MLP.
Yeah, it's all end to end.
Okay. All right. So thank you.
Margarita, just to carry that point forward. So GCN loss and graph loss, is there a significant difference between the two? Because I assume the GCN is being trained just to optimize, the predictions as well. So if the predictions are correct, GCN is being rewarded. If the predictions are incorrect, GCN is being penalized. So is there a huge difference between GCN and graph loss?
The GCN loss is like a classification or regression loss, like we used Huber loss and cross entropy loss.
Okay.
This is for the training of the GCN. Yeah, theoretically, if you have a better graph structure, then you will have better results for the GCN. But we would need to have all of the edges, not a sparse graph, in order to be able to train all of these only with the GCN loss. Because we have sparse graph, we cannot back propagate to all of it, only with the loss of the GCN. We need an extra loss.
And that is because of the, do you think sparsity is affected by your K hyperparameter? Like if you want to make this graph less sparse, you would probably pick larger number of edges.
Okay, the sparsity of the graph is it depends on the number of neighbors we chose for the K.
Yes.
So when we choose 5 neighbors, we will have a relatively sparse graph.
Okay.
We would have had all of the edges, so not keeping only the top K neighbors.
Yes.
And in this case, what we could do would be only to train with the GCN loss, and then we would allocate scores to every edge. This has happened in the literature and it can be done, but then the problem is that you have a graph with many edges, so it's not computationally
feasible anymore. Okay.
So we wanted sparse graph so we can make it lighter, and this is a way to overcome the problem of the sparse edges and to go end to end.
Okay. Okay, so basically, yeah, there's the what I can see. This PIJ is it indicates the link between the patient's I and J, right? In your graph loss, PIJ.
This one.
Oh
yeah,
yes.
this one. Yeah,
Yes,
this is a probability of 2 nodes to be connected or not.
Okay.
It's based on this distance function, which this distance function has the feature phenotypes of the one node and the phenotypes of the other node. So compares how close these phenotypes are. And if they are close, based on cosine similarity, let's say, it gives higher probability. And then when we keep the top K, we keep the ones with the highest probability.
With the highest probability. Okay.
Yeah, and we use the relaxation, the Tom Campbell top K, because the simple KNN didn't really work that well, because I think this relaxation was important, so more edges to be tested, and this gave us better results. So we were not stuck every time with choosing this.
And these that edges are basically directionless edges, right? So they're not directed.
No,
so if there is an edge between PIJ, there is no particular direction. But then is it being rewarded based on only the label of node I and not node J in your loss function, L graph?
Yeah, but this is for all of the nodes.
Okay.
Yeah.
Okay. Yeah, so basically there'll be another term added for the same edge, but with the node J. Okay. Yeah, makes sense. Do we have any more questions from the audience? If not, then let's thank us.
I have one more question if nobody else has. So did you try with you mentioned many graph node GNNs architectures that I think you've worked with. Did you try the graph isomorphic network, the GINs or GINs as they call them?
We have done it. I'm not sure if we did it for only the static graphs or the adaptive. actually, but I don't remember. I would have to check.
And
We tried
yeah,
different.
and in your experiment, you find the GCN to be the best.
No, we found the GCN to be the one that's more like the most affected by the graph structure. So the graph shades work similarly, but it's not affected. It works similarly for everything. It's not. It's like maybe slightly worse. Yeah, so the GCN is better, but it does not improve compared to the static graphs. And we found that the graph shades is quite resilient to the graph structure. So the performance is not affected that much. And we noticed also that every time we trained it here, the attention scores given were not like consistent. Every run, it would give scores to something else for the graph shades, which shows us that it's not affected that much by the edges in the first place. While GCN ended up giving weights to the same phenotypes, like the highest scores to the same phenotypes every time, and there was a huge improvement, like a big improvement in the results. Yeah, but for the gene for the adaptive, I don't remember. I would have to check. Thank you.
So if, for example, graph sage, if it is less affected by the graph structure, that leaves you little room for, interpretability. That's the drawback, right? Because
Yeah.
if it is not assigning the same, it's like consistent weights to the same phenotypes, then it's not very interpretable.
And also there is not much improvement compared to the graph agnostic methods. So
okay.
I would not be sure how to make it work for population graphs all in all. I've read, it makes sense why it works better, because the GCN takes the node and averages itself with all of the neighbors, while the graph shades takes itself and averages all of the nodes and then averages these with itself. So it gives more importance to the node features. So it makes sense why it would work better in a graph structure that's not very optimized, but I don't know how optimized it would have to be to make it work, to make it meaningful to use.
Another thing is that about architecture, like GCNs, that homogenization effect that you were talking about, like if it keeps averaging and you have multiple layers of graph convolution, you end up with basically the same features for everything. That's when it stops being useful. So in your experiments, how many GCN layers you found to be effective? Not many.
Yeah, 3, 2. Yeah, we didn't need much for making it work.
Okay.
If it's too deep, then it gets complicated with the oversmoothing.
Yes. Do we have any more questions from the audience? If not, then let's give our speaker another round of applause. And thank you very much, Margarita. We'll be uploading this talk on the YouTube. Thank you very much, everyone.
Bye.
See you all next week, hopefully.
Bye.
Bye. Thank you.